Welcome to the Explainer. Today we're taking a pretty incredible microscopic factory tour right inside your own cells. You know, when we think of proteins, we usually just think about nutrition, right? Like protein shakes or whatever. But from the keratin in your hair to the venom of a snake, structurally speaking, proteins are the ultimate molecular machines. They literally do almost everything in our bodies, from digesting food to repairing tissue. But they don't just magically appear fully formed. They've got to be built, meticulously, level by level, starting from absolutely nothing.
Which brings us to this really fascinating question. How exactly does a simple line become a complex machine? It's honestly one of the most incredible puzzles in all of biology. How does a flat, basic string transform into a highly complex, perfectly tuned, three-dimensional machine? Well, it doesn't happen all at once. Instead, it happens in four distinct, beautifully orchestrated levels of structural organization. So we're going to unpack the source material today and look at exactly how this transformation takes place, revealing the hidden architecture of life.
Okay, let's dive into this, starting with the very first stage of our cellular assembly line, primary structure, the simple line. At this foundational level, the primary structure is really nothing more than a specific sequence of amino acids. Think of it kind of like beads being threaded onto a string, or, letters being typed out in a long sentence. It is just a flat, simple line. But don't let that simplicity fool you, because the precise order of these amino acid beads actually contains the complete architectural blueprint for what this protein is eventually going to become. It's so precise that if you change even one letter in this sequence, the entire machine could fail. Like, just one wrong amino acid is literally all it takes to cause something as severe as sickle cell anemia. So, how do these beads actually stay together as they come off the assembly line? Well, they're linked by very specific connections called peptide bonds. These links essentially act as the mortar holding our entire foundation together, locking that specific sequence of amino acids into a continuous, unbreakable chain. Without these peptide bonds, our flat, simple line would just fall apart before the real building process could even start.
Once we've got that foundational line securely linked, we move on to step 2, secondary structure, twisting and folding.
Because, as you might guess, our simple flat line doesn't stay flat for very long. The secondary structure is defined by the local twisting or folding of that primary chain. So sections of our long string of amino acids begin to actually interact with their immediate neighbors. They start crinkling, coiling up, and bending into these distinct, localized physical shapes. It's kind of like taking a piece of flat string and watching it start to curl in on itself in very specific, targeted sections.
And from that localized twisting, we generally get two main, highly predictable shapes: alpha helices and beta sheets. Alpha helices look a lot like spiraling staircases, or, tightly coiled springs. And beta sheets? They resemble the folded, zigzag pleats of a paper fan. These two reoccurring structural shapes really act as the fundamental building blocks for the rest of the protein's overall architecture.
But to keep these newly formed coils and pleats locked firmly in place, the protein has to rely on a different type of glue. They're held together by hydrogen bonds. While our foundational peptide bonds held the actual line together, it's these hydrogen bonds that secure the folds. And there are just so many of them forming across the chain that they provide this immense stability, keeping those alpha helices spiraled and those beta sheets pleated so our assembly line can keep moving forward.
Right, so we've got our local folds. But obviously, a few coils and pleats don't make a functioning machine. That brings us to stage 3, tertiary structure, the total 3D shape.
Up until now, we've really just been looking at localized neighborhoods along the chain, but the tertiary structure represents the grand, overarching, total 3D shape of a single protein chain. This is where all of those alpha helices and beta sheets we just talked about fold in on themselves again, crumpling into a very precise, totally specific, and highly complex three-dimensional object.
Now, what's really interesting here is exactly how that total 3D shape is determined and held together. It's actually formed by side chain interactions. See, every single amino acid in our primary sequence has a unique side chain hanging off of it. And at this tertiary level, these side chains start vigorously interacting with one another. It's like this complex molecular dance where these unique side chains push and pull on each other. Some are attracting, some are repelling, and that push and pull forces the entire chain to collapse into its final, intricate, and totally unique tertiary form.
Now, for some proteins, that 3D shape is the end of the line. But for our most vital molecular machines, there's one last step. Step 4, quaternary structure, the functional unit.
At this quaternary level, a single 3D chain just isn't enough to get the job done.
This massive structure is made when two or more distinct protein chains join together. Imagine taking several separate, incredibly complex 3D jigsaw puzzle pieces and then locking them all together to create an even larger, multi-part megastructure. Hemoglobin, the protein that carries oxygen around in your blood, is actually a perfect example of this.
So the crucial point is that this combined multi-chain structure represents the final functional unit. This is the finished working product finally rolling off the biological assembly line. It's only at this stage, when multiple tertiary structures have bonded together, that the protein is completely finished and fully ready to perform its specific task within the body.
Okay, let's move to our recap and see how this builds together into a complete journey, because it really is an awe-inspiring process. Step 1 is the sequence, the primary structure, that flat, simple line of amino acids. Step 2 introduces local twisting and folding into those secondary helices and sheets. Step 3 collapses all those twists into a complex, overall 3D shape, the tertiary structure, based entirely on side chain interactions. And finally, Step 4 brings multiple folded chains together into a working quaternary unit. Just 4 steps, transforming a flat line into an absolute biological marvel.
Which brings us to the end of our explainer and leaves us with a truly mind-bending question to think about. If just one fold fails, does the entire machine break? As we've seen today, ultimate biological function relies entirely on the precise execution of every single one of these 4 structural levels. A single mistake way down at the primary sequence can cascade all the way up, completely altering the folds and basically ruining that final functional unit. It's a delicate, beautiful, and just incredibly precise balance. Thanks so much for exploring this amazing microscopic architecture with me today, and keep questioning how the smallest building blocks create the most complex machines we know.